Let's move along then to Cesar Garcia and talking about one of his company's tests and some broader implications. Thank you.
Thank you, Andy. Good afternoon, everyone. I'm Cesar Garcia. I'm the Chairman and CEO of Iris International. And probably you know our company more for diagnostics urinalysis and personalized diagnostics. Iris is the market leader in urinalysis in the United States, so most likely every time that you go to your doctor and they perform a urine test, it's performed by one of our instruments. But today, I really want to talk to you about our personalized medicine initiative, and that hinges about a technology that we call NADIA, nucleic acid detection immunoassay, and our first application is for a prognostic prostate cancer marker. Moving on in terms of what is a prostate cancer problem. Prostate cancer is a very serious health issue. There's about 220 to 250,000 new patients discovered every year. People die from prostate cancer. The global incidence is about 700,000, so you combine that, it's about 1 million men suffer from prostate cancer every year. The treatment options are all significant. It's basically surgery, the radical prostatectomy or the removal of the prostate. It's external beam radiation therapy, radioactive stick implantation, or active surveillance, which doesn't mean to let the patient with no treatment. It means that the doctor decides to monitor the patient very closely until there is a reason to initiate treatment. 95,000 men every year go through a radical prostatectomy. And the number is increasing because of the early detection of prostate cancer, and also because the availability of minimal invasive robotic surgery, basically the Da Vinci robots that were commercialized by Intuitive Surgical. So a urologist decides to perform a radical prostatectomy when prostate cancer is confined to the prostate gland. And typically, what you discover that by monitoring the prostate-specific antigen, performing a biopsy, or following the patient's symptoms. So let's say that the patient decided with the doctor to go for a prostatectomy. After the prostatectomy, the prostate is removed, and there is an anatomical evaluation, a pathological evaluation of the prostate tissue. So they look into the Gleason score. They look into the disease of the level, how big the tumor was, the spread of the disease, the pre-PSA level prior to the radical prostatectomy. They monitor the level of PSA, and based on that, they classify the patient between high risk, moderate risk, and low risk. To be candid with you, the issue of treating the low-risk patients is easy. Most of them are cured. Prostatectomy is very effective in cure prostate cancer, or 50% of the men are cured. The issue is the other 50% that are in the moderate to high-risk category. The doctors don't know what to do. The patients are very concerned because they were classified to be medium to high risk. And up to the release of Nadia Prosview this year, there was no diagnostic test to help them make that decision. There was an independent study performed in 1999 by Dr. Pound, and they took a cohort of patients that were not treated. These patients were classified to be low risk, moderate risk, and high risk. You can see the problem here. Of the 50% that were moderate, only 23% of them have biochemical relapse. Unfortunately, more than 50% of those patients, they receive today radiation therapy or androgen deprivation therapy, which is not needed. Even on the high-risk profile, 63% relapsed, 37% did not relapse. As you will see in the presentation, Nadia Prosview has a capability of helping doctors and patients to make that decision. We don't claim that Nadia Prosview is the only way to do it, but it's really a significant tool to make that decision. So what we did about 10 years ago, we began the development on an ultra-sensitive and very precise prostate cancer marker using the combination of two technologies: immunoassay to capture the PSA, which is a protein, the prostate-specific antigen, and real-time PCR to do the amplification. So Nadia combines immunoassay with nucleic acid, and that's what we call it, nucleic acid detection immunoassay. It basically provides an individual decision point for every patient by every doctor. For the patient, it provides the clinical clarity for the patient at high risk. They don't know what to do with these patients when they're in moderate to high risk. Treatment of prostate cancer post-prostatectomy is a financial burden to the healthcare system. And also, one of the problems, big problems with prostate cancer treatment is not only the disease, it's lack of peace of mind. The man is very concerned because of the disease and the fear to die from prostate cancer. Now, what we did in Nadia, basically is an ultra-sensitive protein detection system using the combination of immunoassay and real-time PCR. And in traditional prostate-specific antigen testing, you may get a value of 0 or close to 0. But 0 is not really 0, it's the limit of detection of the assay. So what we did when you're counting in traditional immunoassay, you're counting probably to about 100,000 molecules. Nadia PSA can count down to single molecules. We also tested for the FDA, because it's really an assay cleared by the FDA. We tested the linearity and the precision of the assay. And if you see, the dynamic range of the assay is very high, very linear, so it's very sensitive and very precise. And I will explain to you why that is important. Nadia ProSview was cleared by the FDA in October of 2011. It's not a lab-developed test. It was developed with the rigor of any other diagnostic test. And the FDA required that to release Nadia ProSview, we have to confirm the recurrence of prostate cancer against a hard clinical outcome. So we're not confirming here that we're not trying to predict the elevation of PSA after prostatectomy. We're trying to identify that the patient truly relapsed by finding cancer in the imaging, in the biopsy results, or death of prostate cancer as a prostatectomy. How does Nadia work? Well, we take 3 samples, 3 blood samples, one 3 months after the prostatectomy, one 6 months, and one 9 months after the prostatectomy. We plot the 3 points, and we look into the slope of the 3 points. Now, very important, to be able to plot that line, you have to be extremely precise, because if you repeat the same patients and you get a cloud of points, you get different results, you cannot draw a line. To give an idea of what we discovered is that this man, stable men, they have a concentration down to about 2.5 picograms when the limit of detection of traditional immunoassay reliably is about 200 picograms. So we have to be about 100 times more sensitive than traditional PSA and very, very precise. So when we plotted 3 points, we basically developed a hypothesis, and the hypothesis was proved by the clinical study that we performed. The hypothesis was that a man that has an elevation, a rise of PSA of less than 2 picograms per milliliter per month, that patient is stable, and that patient does not need to follow up treatment with radiation therapy or androgen deprivation therapy. If the patient is not stable, that patient must be followed. So the official claim that we got from the FDA was that Nadia ProSview is indicated for use as a prognostic marker in conjunction with clinical evaluation as an aid in identifying those patients at reduced risk for recurrence of prostate cancer for 8 years following the prostatectomy. That's a very strong claim supported by clinical data, and to our knowledge is the first time that FDA approved a marker to diagnose stable patients after a prostatectomy. So our proposition is don't treat patients that don't need treatment. And that's a very important decision, it's a critical decision, and that's why the FDA took us through a very, very rigorous testing. The clinical study was very significant. We have to search for 27,000 patients. These are retained samples. Of the 27,000 patients that we sorted, we found about only 700 that qualify with all the criteria that the FDA required. We selected on a random basis about 300, and these are the results of that random study. Remember that this is not about predicting biochemical recurrence, it's actually confirming that prostate cancer was present in bone scans, in biopsy, or death from prostate cancer. The results are the following. We achieved 92% negative predictive value. So when you said that the patient is negative, 93% of the time we were correct. It's unusual to see a prostate cancer marker with a very high positive predictive value. Not only we achieved a 93% negative predictive value, we also achieved a 78% positive predictive value, which is very important for the doctors, because when you tell the doctor that patient is not stable, you're saying that that patient is positive. They don't want to be treating false positives. So the balance between the false negatives and the false positives is very important. Now, the FDA asked us to do something more. Try to do an analysis of those samples and correlate the slope to the actual recurrence. So we went back to the data, and we plot the Kaplan-Meier plot that basically plots the probability of being stable after a number of years of surgery. The power of Nadia Prosview is such that you see the green curve in there, 90-some% of the men were stable for 17 years. And 17 years was the limit of the samples that we collected. Probably were stable forever, but we don't know that. And we cannot make that claim because the data doesn't support it. But what is very telling is a separation between the two curves. When Nadia Prosview said that the patient is not stable, the probability of relapsing or having a clinical relapse drops tremendously in 5 years. The probability of being stable is only 50%. So when you compare the two groups and you compare the curve, basically the patients that were identified to be non-stable, practically all of them relapsed over the duration of the study. So the clinical benefits are many. It provides an objective tools that enable the clinical clarity to treat moderate and high-risk patients. It provides a tool for the doctors to optimize the management strategy for the men post-prostatectomy. Not only we want to optimize the clinical value, we want to optimize the clinical value by avoiding radiation therapy, androgen deprivation therapy, and to minimize the morbidity associated with prostate cancer treatment, which is typically sexual impotence or urinary incontinence. It provides peace of mind to the patients, and it provides also the potential that men that are cured, that they have an objective measure that they can go to their insurance company and say, I can be insured because I'm cancer-free. Today, they don't have a way to get that. And conversely, the men that are cured, but obviously they suffer significant sexual dysfunction and they need testosterone therapy. The doctors do not prescribe testosterone because it can increase the tumor again. But if they're cancer-free, they can take that therapy and resume a normal life. So approximately 25% of the patients post-radical prostatectomy receive radiation therapy or androgen deprivation therapy. It's very expensive. Radiation therapy costs about $50,000 per patient. That doesn't include the cost of treating the comorbidities that I mentioned before, urinary incontinence, sexual dysfunction, bladder and rectal cancer, proctitis. Androgen deprivation therapy, we come to about the same number because over 3 years is about 48,000 different type of complications. The cost of treating prostate cancer in the United States is about $2.5 billion a year. Our estimates are that if Nadia Prosview was adopted as a treatment paradigm, we could save about $1 billion to the healthcare system by avoiding unnecessary treatment. The path to getting this clear has been a long path. We began the discovery about 10 years ago with a patent from the University of Berkeley, California. We went through the FDA for a long process, about 3.5 years. The assay was cleared in September and was launched in March. And it's very important to mention this because what happens is we in the AdvaMed members companies, we go through a very rigorous development process. I can tell you now that there's already lab developed tests that are claiming to do the same, and they haven't gone through that. And many of them, what they're doing, they're claiming biochemical recurrence when the FDA clearly says that's not an indication of prostate cancer recurrence. Now, you have the 510K clear, you went through that rigorous process, it took you 10 years, $35 million were spent. We're not there yet. The second part is to get reimbursement. And right now, we're going to the market. The company's funding the utilization of the assay. The assay is expensive. It costs about $500 just to collect the samples. The price of the assay will be in the order of $3,000 to $3,500 per test. And with their first approach to get reimbursement, we're going to be addressing the integrated delivery networks, Kaiser Permanente, Mayo Clinic, the Department of Defense, the Veteran Affairs Administration, and in parallel, we're working with CMS to get reimbursement. We require analytical papers. 2 of them are already published, clinical papers. 1 of them is published, the other one is in process, and we have a field experience trial, which is designed to prove the impact in patient treatment and the economic benefit of using Nadia ProsView to treat patients. Based on the actuarial analysis that we have performed, we believe that the savings to the healthcare system will be in the order of $10,000 to $15,000 per patient. If you avoid unnecessary treatment, that's why we price it at $3,500 so we can recover our investment and we can fund new technologies. So we expect to get reimbursement by 2013. Now, count when we began the process. It's going to take us 10 years to get through the discovery and the FDA process, another 2 to 3 years to get reimbursement. In that time frame, we could have been saving money. And this is really brings me to the conclusion of my presentation. There's a general idea that diagnostic tests increase healthcare costs. I don't believe that. I think that effective target therapies, effective target diagnostics, they have not only the potential of improving patient care, but they have significant potential to reduce healthcare costs. And like I said, in this case, $2.5 billion a year spent, we could save probably $1 billion. What we need improvements to the regulatory and reimbursement systems, better coordination between the agencies involved, so they can provide the support and expedite the implementation of this kind of innovation through the diagnostic industry. So that concludes my presentation. I can take any questions from you. Thank you.
Any pushback from the urologic community relative to the subsequent samples that have to be taken, and quite possibly the patient community as well? And what are your thoughts about the morbidity there? I assume you're going back to the OR to do scoping and do procedures, correct?
Yes.
Any thoughts about morbidity there as well?
So the good news is that the test is a blood test. So we don't have to go back and check the patient. It's a blood test. The reception from the urologists have been excellent. We have many of the luminaries in prostate cancer are in our scientific advisory board. We began already testing in Johns Hopkins. We have talking to Kaiser Permanente, the Department of Defense. So the reception has been very good. Actually, one of the biggest concerns was the radiation oncologist, because we thought that we were going to basically get a pushback because obviously immense revenue for them. And the reality is radiation oncologists, they do not want to give radiation to a therapy that doesn't need it. And conversely, sometimes insurance companies do not approve radiation therapy when a patient needs it and they don't have an objective indicator to initiate radiation therapy. ProsView can help them in both ways. Yes. Thank you.
I noticed something really interesting in the beginning of your presentation,
Yes.
which is sort of related. I noticed you put a statistic about how many occurrences there are of prostate cancer in the U.S. and then around the world.
Mm-hmm.
And I noticed that in the U.S. that's a huge sum. You have 228,000 out of the global incidence. That's almost a third.
Yes.
So for me, in my mind, that probably spells some kind of environmental factor. Have you studied?
I think that it has to do more with the treatment paradigms. Remember that when you talk the total population, if you go into rural China and India, and you can't use those countries to count in the population because unfortunately, those people don't have the access that we have here. So, but the presence is very prevalent. There's about 1 million men that survive over the last 5-6 years. So then 20 million men surviving prostate cancer, and that will be one of the follow-up applications to actually monitor those patients. We don't have that claim yet. Yes, sir.
Why CMS national coverage analysis rather than local carriers?
We're going to do both ways, okay? We're going to begin with Palmetto, okay, for California. But it's important to really address it in parallel. So the answer is we're going to go both ways.
No, CMS has a reputation for not liking when, CMS has a reputation for not being real fond of companies going parallel. I mean, are you not worried about a little bit of backlash from Louis Johnson?
We believe that we're going through Palmetto first, and then following with CMS will be the route. But one of the things that is important to keep them kind of informed of what we're doing, we believe that it's kind of opening the dialogue will be but the first priority will be Palmetto.
So you're going to Palmetto?
Yes.
First,
Yeah.
and then
Then CMS, yes.
so it's not parallel.
Yeah. This is staggered.
Great, thank you, Cesar. I know
Thank you very much.